Last part of muscles. We've been working on skeletal muscles the whole time. I just want to briefly compare skeletal to cardiac and smooth. It's not outrageously difficult or anything, but the things that they have in common versus different. So this is, of course, a skeletal muscle fiber, right? And so what does that have in common with cardiac muscle, and what does it have different from cardiac muscle? Well, cardiac muscle is striated, so it probably won't surprise you that it actually has sarcomeres inside it that are really, really similar to the sarcomeres that you have in skeletal muscle. And so the striping comes from the sarcomeres. So it's striated, skeletal muscle striated. It's involuntary, though. It's not controlled by the voluntary division of the nervous system. And it has the same arrangement of actin and myosin. The fibers are shorter. Remember, a skeletal muscle fiber can be up to 1 foot long, but cardiac, you can usually see the boundaries of a cardiac muscle cell in a single slide. So shorter fibers, 1 nucleus per fiber instead of being multinucleated. And remember that the fibers generally branch. And the other thing is the intercalated discs have gap junctions, which means that if I tell this cell what to do, it can actually tell its neighbors. So that's different than skeletal muscle because you don't need to tell every single cardiac muscle cell to contract. Because if you tell one, and they can actually spontaneously initiate it themselves, then they can tell their neighbors. So each fiber does not require individual innervation. And then smooth muscle, which is last, just to compare. Smooth muscle is found all over the place in the walls of hollow organs and hollow tubes, blood vessels, that kind of stuff. It's non-striated, which might make you think, oh, it doesn't have actin and myosin. It does. It's just that they don't line up, so you don't get stripes. It's also involuntary, like cardiac muscle, unlike most skeletal muscle. Now, notice that this kind of looks like actin and myosin, doesn't it? Those little pokey-uppy things. And then the myosin. But if you don't line up all of the myosin, you don't get a stripe. So instead of just pulling together this way, it pulls together a bunch of different directions. So it still kind of has a sarcomere-like thing. It's not called a sarcomere. But since they don't line up, you don't get any striping. It still has actin and myosin, and it still works with the sliding filament theory. Okay, the cells are much smaller. They're spindle-shaped, and they're uninucleated like cardiac muscle was uninucleated. They do contain actin and myosin, but the filaments are not so orderly, so there's no sarcomeres and no real striations. And so what happens inside hollow organs and tubes to make things contract is that the smooth muscle, if this is a tube, smooth muscle fibers will be organized in a circle right next to the lumen of the tube, and then superficial to that, they'll be longitudinally arranged. And so in order to get things to move through the tube by peristalsis, the circular layer will contract and it will squish, and then the longitudinal layer will pull and it will move through, which allows these to sort of cause peristalsis as you squish, pull things through a tube. So let me just show you this weird little video. Hopefully it will work. That shows peristalsis actually occurring. This is somebody's guts. And if you give it just a second, you can actually see peristalsis moving through. This is endoscopy. Peristalsis moving through the gut. Give it just a second, you'll actually see it. So you see things actually moving through the gut as you squish and then pull and then squish and then pull. I don't know why my email just opened up. Okay, so that's it for muscle, and we will cover what you guys get confused on in class, so don't feel shy to ask. You don't need that. Okay,